Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Biological Macromolecules, Exploring Analytical Development and QC Case Studies in Antibody Developability and Biologics CMC. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank ChemPartner, who developed the content for this presentation. Over the past 20 years, ChemPartner has evolved from the pure chemistry service provider to a research innovation engine with a broad portfolio of services spanning from discovery through development and manufacturing in both small and large molecule therapeutics. Shanghai ChemPartner, which includes ChemPartner and ChemPartner Biologics, offers a broad range of drug discovery, development, and manufacturing services, including discovery chemistry and biologics, biology, pharmacology, DMPK, and exploratory toxicology. Now I'd like to introduce our speakers for today's event. Dr. Chin Sun worked in biomedicine and clinical biotechnology for many years in the United States. He has technical expertise in preparation, purification, and characterization of biological macromolecules, with a focus on analytical development and quality control and execution of analytical methods for pharmaceutical raw materials, drug substance, and drug products of biopharmaceuticals, including antibodies, recombinant proteins, fusion proteins, antibody-drug conjugates, polypeptides, and vaccines. And Dr. Jinghan Su researched targeting drug delivery systems based on nanotechnology in Shanghai Institute of Materia Medica until 2017. She has published 10 papers on advanced materials, advanced functional materials, theranostics, biomaterials, and more. In 2017, she joined ChemPartner Biologics, where she leads the research and development of biologics products and has successfully led or supported over 30 biologics formulation and process development projects. But now, without further ado, I'd like to hand the presentation over to our first speaker today, Dr. Chan Su. You may begin when you're ready.
Thank you very much, Ryan. Thank you everyone to join us our webinar for the strategy for characterization of protein biopharmaceuticals. We are from the ChemPartner Biologics, Shanghai, China. The first slide, we were talking about the macromolecules. The four major macromolecules are carbohydrates, lipids, proteins, and nucleases. The biopharmaceuticals includes the monoclonal antibodies, recombinant proteins, fusion proteins, antibody-drug conjugates, polypeptides, and vaccines, and more. So unlike the small molecular drugs with rigid structures, the macromolecules undergo complicated folding and can exist in a number of low-energy structures, which poses a challenge for traditional methods in structural biology. The appropriate and complete analysis and characterization are important to the control of their safety, purity, potency, stability, and overall quality. So for the biopharmaceuticals, the powerful therapeutics for the characterization has drawn considerable attention and urgency. So the characterization of biopharmaceuticals is challenged and utilize the state-of-the-art technology to meet international harmonized guidelines Q5E and Q6B. The ICH Q6B document provides guidance for established specifications applicable to commercial biological products. The ICH Q5E is focusing on the comparability of biological products subject to changes in their manufacturing process. So the structural and the physical chemical characterization are a crucial part of biopharmaceutical product development. These are some of the analytical methods and tests that allow drug developers to be sure that they have correctly manufactured their target drug. So it's very important to begin characterization studies at the early stages and scales of product development to ensure that the product and the manufacturing process are correct from the outside. So from the first step of the process characterization, we see the analysis of the media components for the cell structure. In the fermentation step, the cell line characterization will be used. In the purification steps, we analysis of ligature. In the drug substance, we are testing the host cell protein, the host cell's DNA radius. So for drug substance production, we always perform the product characterization, including the primary structure, the secondary structure, the tertiary structure, and the function analysis, the product linked variation, the formulation stability, linked modifications. So now we are talking about in the detail. So the FDA and the EMA have set their own guidelines for the protein characterization methods. So both agencies have adopted the characterization approach outlined in ICH Q6B. So the approval of monoclonal antibody-related products is dramatically increased in recent years. So among them, monoclonal antibody are the major product, which consisting of 77% of the total. After the first approval of a full-length monoclonal antibody in 1998, monoclonal antibodies are the major product in the biopharmaceutical industry. So the monoclonal antibody products, which always include the Fc fusion, Fab, ADC, and bispecific monoclonal antibody bispecific T-cell engager. The new analytical technology and modifications to existing technology are continuously being developed and should be utilized when appropriate. The degradation of a monoclonal antibody can negatively affect the product quality, safety, efficacy, and thus need to be detected if it occurs. In right panel, we see the arrows indicate the main degradation site. The commonly used methods that detect the degradation products are shown in the here. For example, the activating isomerization, which we are using the CX, CIF, IF, LCMS, SPEC to perform the analysis. The most commonly observed degradation pathways are aggregation, fermentation, deamination, and oxidation. On the specific conditions, limited degradation pathways could become dominant. For example, the high pH conditions can take disulfide forms related degradation pathways such as the formation of cider or covalent aggregates due to the disulfide bond scrambling. Incubation with reducing sugars resulting in glycation of many the surface exposed lysine radius. So for in this characterization, the technical selection will be always the question. Appropriate group of selected technicals can be applied at the outside of the product development side, which like the molecular weight analysis for product intactness, the peptide mapping for basic sequence analysis and assessment of translational modifications, the glycosylation, population analysis for glycoproteins, the aggregation analysis using SEC mode and or SVR, the basic higher order structure to check the folding is correct or not. The structure characterization methods should also be used to assess the product following any significant change in the manufacturing process. So as part of our protein mass spec services, we provide a peptide mapping service that allows a client to meet the structural characterization requirements of FDA, EMA, and NAPA, understand the primary structure of the protein, and also assess the post-translational modifications to confirm the genetic stability, to demonstrate the integrity of disulfide bonds, to make key development decisions. Now we are talking about the peptide mapping. The peptide mapping was carried out by digesting protein samples with endoproteins, such as the trypsin, and the subsequent separation of peptide fragments by IBHPLC prior to protease digestion. The denaturation of test protein with known concentration is needed to ensure the complete digestion. Any variations in the mass can be investigated to determine their cause. So the analytical methods provide detailed information of primary structure for given protein and enables the control of the protein sequence down to the level of single amino acids by coping with the mass spec. So in this figure, we see this is the data obtained from the online LC-ES mass spec analysis of protease digest of a monoclonal antibody reference product and 3 batches of the biosimilar. The biosimilar batch 2 and 3 differ from the reference product around the 12 minutes mark. On the 12 minutes mark, there is a difference about the two figures. The peptide mapping can be used to investigate the structure of these components and, if necessary, the orthogonal tests such as the ICIF can be performed to corroborate the reasons for differences in the peptide mapping data. We are using the LC mass spec to check the detail of the information. So this information based on the figure on the left panel, we can see this figure shows the ideal for the intact proteins and peptides, which presents the very high accuracy and high dynamic range. So this is our instrument of the LC-MS spec. So it has the gold standard off-trap detector to provide the optional intact protein model. About the peptide mapping applications, so it's usually for the sequence confirmation, glycosylation, and terminal pre-agglomeration, the disulfide bridges, the heavy chains, C-terminal lysine, deamination oxidation. Based on the sequence of the target protein, the overlapping peptides will be sized as 15 or 12 moles, which covers the full sequence of our protein. So the peptide library is immobilized on surface of a sensor chip, and the analyte molecule is injected in the solution through the flow cell, so which we are using the SPR spec. So the SPR spec is a rapidly developing technique for the study of the binding events involving the proteins which are the major molecular targets for the validated drugs. So for the left panel, we can see through the flow cell analyte. By monitoring the change in the intensity of the reflected light caused by the change in the reflectable index, the peptides are screened against the analyte protein, and those can associate with the analyte are identified, which correspond to the regions of the target protein where binding occurs. For the protein glycosylation, so glycosylation is one of the most important PTMs to consider during protein characterization, and it's a regular requirement to characterize the glycans on biopharmaceutical. So the glycosylation is defined as the enzymatic attachment of carbohydrates or sugars to protein backbone. The two most common types of protein glycosylation is N-glycosylation and O-glycosylation. The N-glycosylation sites can be predicted by the sequence aspirating X and serine, and whereas for the N-like glycans, where the nascent protein enters the endoplasmic reticulum, a block of sugars is transferred into the animal group in the aspirating side chain. With the increasing application of the protein therapeutics such as the monoclonal antibodies, glycoproteins, routinely monitoring the N-glycosylation of proteins gets popular. The right panel is the types of N-glycans produced in different organisms. The role of the protein glycosylation is about we know that many proteins are modified with glycans. Indeed, the majority of proteins on the market today are glycoproteins, which include the monoclonal antibodies, erythropoietin, the W protein, FSH, and other drugs. The fact that so many biopharmaceuticals are glycosylated make glycosylation one of the most important PTMs to consider during the protein characterization. For the N-glycan analysis, the ICH-Q6B guidelines require full characterization of the glycosylation of a glycoprotein. This applies to both the total glycan population, but also the structures of the glycans at the individual glycosylation sites if more than one is present. The aim of this set analysis on the left panel is elucidate the global N-glycan profile of a molecule of interest. So usually the steps including the denaturation, deglycosylation, purification, labeling, and the SPE purification. Most of the N-glycan sample preparation consists the general steps including digestion of the glycoprotein, deglycosylation, purification, labeling, and the SPE. So the left panel represents a high we know the mass spec represents high sensitivity and robust for the structural characterization of glycosylated compounds, and can also be used in the high throughput manner. The N-glycans released by the enzymatic deglycosylation using the peptide Sidase A or S is analyzed after polymerization on the left panel. We also use the Helix UPLC mass spec for analysis in analyze the N-glycan profiling. The Helix has been a powerful technology for the N-glycan profile. So before the due to its wide acceptance, the glycan profiling of enzymatic glycan release with PNGS F followed by the devaluation by 2AB by Helix UPLC is a fast and robust technique. Before the fluorescent labeling, the harvested the N-glycans should be further purified on a TSK column when large quantities of detergents. So the labeled N-glycans are then analyzed via the Helix separation combined with mass spec detection. So for the biopharmaceutical characterization, we also talk about the high-order protein structure analysis. So the high-order protein structure is responsible for the correct folding and the three-dimensional shape of a biopharmaceutical. These can be affected by different formulations, which in turn can affect protein activity. The folding and the shape of the protein impact directly on the functionality of the drug, and this correlation is often termed as the structure-function relationship. We are using the circular dichroism to check the secondary structure and the tertiary structure. You know, if a sample is too concentrated or the patch length is too long, the sample will absorb too much light. Not enough will pass to the detector for a reliable CD signal to be required. We can see the right side panel. Conversely, if the sample is not so concentrated enough, or the cell path length is too short, the CD signal will be too weak or non-exist. So usually what concentrations and the path lengths are necessary to obtain the good CD data? So for the secondary structure studies, usually 0.1 milligram per ml of a sample in a 1 millimeter path length cell is recommended. The water becomes optical in a 1 centimeter path length cell below 185 nanometer, and the medic buffers and salt will absorb too strongly in a 1 centimeter cell to acquire the CD signal below 200 nanometer. For tertiary structure studies, 1 milligram per ml of sample in a 1 centimeter path length cell is required. After passing through the sample, the left and right beams have different amplitudes, and the combination of the two unequal beams gives elliptically polarized light. We can see the right side panel. Means the CD measures the ellipticity of the transmitted light. A chiral molecule is one with the low degree of symmetry which exists in two mirror image isomers. Isolated image is an example of a CD in glucose. So between the two circles is a glucose, a simple soup. So this is this figure, the upper panel is for the CD or workflow. The B panel is a CD spec of the characteristic secondary structure model detected in the proteins and peptide. You can see on the red line is a helix structure. The black line is a sheet structure. The green line represents the random coil. The C panel is about the near UV CD spectrum of two proteins, one containing the tryptophan and tyrosine, and the other only the tyrosine, on the black line. So the far UVCD data obtained from the analysis of RMP and biosimilar of the monoclonal antibody. So the RMP means the reference medicinal product. So you may ask me, what conditions favor deamination? So let me talk about the deamination afterwards. So here in this panel, it suggests that the secondary structures of these two samples that analyzed are comparable because they are overlapped. So for the deamination is in point PTM type. So the protein PTM are common to the biologic drug substance produced by the bioprocessing. The PTM analysis mainly refers to identify the modification site, modification type, and the relatively abundance of each modification. For the peptide sequencing, we usually use the tandem mass spec. The tandem mass spec provides the mean for the amino acid sequencing of peptides. So the selected peptide ion species is activated after the peptide ion of interest is isolated. The mass to charge values of the fragment ions are determined in the right panel. So in this panel, it's a data from a native protein and the deamination protein. So what conditions for favor the deamination? So we know the highest frequency in the protein is with the asparagine and the glycine sequence. So the intermediate frequency in asparagine reduced, followed by a polar amino acid with a relative small side chain, is like the serine, threonine, aspartic acid. So in this panel, we can see the differences. The differences for the deamination after the asparagine deamination into the aspartic acid results in an increase in mass of a single daughter. Our company, Component Biologics, in Shanghai, China, delivers a one-stop service that ensures the efficient and economic return to customers' requests for biopharmaceutical analysis and characterization. We take pride in providing the optimal support, and we wish to build the long-lasting partnerships with our customers. We provide the full service solutions from the analyzing and the evaluation the quality of biopharmaceuticals from pre-formulation to commercial manufacturing. Our CMC and GMP analytical services includes the GMP release tests ranging from the physical, chemical, and bioactivity analysis, microbiological testing, and analysis of biopharmaceuticals with FDA, NABA, EMA accepted quality management system. So we also have the service for the analytical method development, of optimization, validation, verification, in-process sample testing, DSDB release testing, and stability studies. So always, the state-of-art service and support has always been our priority. So thank you very much, everyone. So if you have any questions, you may send me the email and talk with me. So now let me pass to my colleague, Dr. Jinghan Xu. Thank you.
Hello, everyone. I'm Jinghan Su, and I'm in charge of the research and development of the pharmaceutical department in ChemPartner Biologics. It's my honor to speak here, and today I will be very glad to share you something about antibody developability assessment. This slide shows the agenda of my presentation. Firstly, I will introduce the background of antibody developability assessment. After that, we'll take a look at the general workflow of the experiment. Then I will show you some typical cases we encountered in our work. And lastly, there will be an introduction of our company. I hope this content can be helpful for your work in antibody development and developability assessment. So why we need developability assessment? As we all know, most biotherapeutic candidates will fail during what have been termed the valley of death or the translation gap. This is due to the long duration of each development stage, capitalized costs, and the probability of failure, which tends to accumulate in later development phases, especially between phase 2 and phase 3. According to statistics, every 250 compounds entering preclinical development, there's only one who finally receives the regulatory approval. The failure is caused by many aspects, including the stability and quality issues, the efficacy shortcomings, or even the safety problems. And we found that all these are closely related to the design of the molecular structure, the selection of the leading candidate, and the development of the process. So our scientists consider that if we introduce some key evaluations at an earlier stage, such as can it be made, is it safe, or will it work, then we could know more about our candidates and allow to change in earlier life cycle if we need it. All this can reduce the risk of drug development. And this approach is called developability assessment. So, what is developability assessment? Firstly, it is a multidisciplinary approach. We will use a variety of investigation methods to characterize our candidates. Besides, it's a ranking tool. When we have several candidates, we can compare this data and identify the leading candidate with less risky at an early stage. On the other hand, it is also a two-way communication bridge because it can connect discovery and the CMC development functions more closely, which enable a smooth transition from discovery to development of the leading candidate. Finally, it is also a solid foundation for CMC development. With a systematical evaluation of the candidate and the knowledge of any remaining identified risks, the failure of CMC development will be greatly reduced. This slide shows the major components of developability assessment, including safety, structural assessment, and manufacturability. We can see that there are many aspects of each field, such as PK, PD, in silico hotspots analysis, the cell line stability. Among them, the structural assessment is the most important part because it will impact the candidate's behavior in the other two aspects. This slide shows the workflow of the antibody development, and we can see that the developability assessment is usually integrated in two phases. Firstly, the antibodies are typically generated by immunization and screened by functional assays. So during developability assessment 1, we access predominantly the CDR regions. Then the candidates will be humanized or re-engineered and go through a second round screen. So developability assessment 2 examines the whole molecule. So next, I will show you a general workflow of the developability assessment in our company. This slide gives an overview of our proposal. Since the antibody developability assessment is highly personalized, so in the actual project, the experiments will be added or reduced according to the client's demand and case by case. In phase 1, we will do an in silico sequence analysis, and it will cost 2 weeks. Then we will do the preliminary physicochemical characterization of the candidate, and it will cost 2 weeks, and the sample amount is 3 milligrams per candidate. If the PTM analysis is necessary, another 2 weeks is needed. In phase 3, we will do the preliminary stability study, and it will cost about 9 to 11 weeks. Besides, for the subcutaneous and intramuscular injection candidate, since the protein concentration is usually very high, so an additional high concentration protein stability study will be needed. In the following slides, I will give a detailed introduction in each phase. This slide shows a picture of the common degradation sites of the maps. We will conduct the homology modeling of the FV region based on the sequence to check if there are some potential hotspots. If necessary, we can further do some stressed experiments to check the existence of these spots by PTM analysis. In phase 2, we will carry out some preliminary physicochemical characterization of the candidates, such as the molecular weight, the melting temperature, the aggregation temperature, the bonding to characterize their structure, the conformational stability, the colloidal stability, and the affinity. If there are more than one candidate, this data can also help us to do a preliminary stability candidates ranking. In phase 3, we will conduct a preliminary stability test of the antibody. Firstly, we will generate the protein samples and evaluate its manufacturability. The protein will be expressed in multiple cells, and its titer and the purity will be tested. After that, the antibody will go through 2 to 3 steps of purification, during which the yield is calculated and the in-process stability is monitored. During this process, the candidates with low expression level, low recovery level, or low in-process stability will be excluded. After the protein generation, we will carry out the preliminary stability studies, which usually involves the following 3 steps. Before the experiments, the sample will be firstly transferred to a suitable buffer system. In the thermal stability study, the samples will be placed under different temperature conditions. The forced degradation conditions include the free sol stability, high pH, low pH, photostability, and if necessary, the oxidation and the glycation experiment will also be conducted. For the candidates with high protein concentration, an additional stability study will be needed. We will test their solubility by a PG precipitation method, and we will also test their viscosity and do some preliminary stability studies. This slide shows a risk assessment table for the preliminary stability study, which was concluded according to our previous work and experience. For example, if the isoelectric point of this molecule is more than 8, we can say that in this aspect, the risk is low. Besides, for the change of the size variance and the charge variance, there are also corresponding accessing standard. This slide is for the stressed experiments. For example, in the high pH experiment, if the change of the monitor percentage tested by SEC-HPLC more than 2%, it may suggest that the high pH condition is of high risk for this molecule. So in these years, we have worked on a lot of projects about developability assessment, and next I will show you some typical cases. This is case 1. We conduct a developability assessment study for the MAP-A molecule. In the left figure, we test the free self-hydroid groups percentage, and the value is 0.26, which suggests a low risk of free self-hydroid groups in samples. The right figure shows the melting temperature testing, and the result is more than 60 degrees, which suggests a high thermodynamic stability. Then we also conduct the freeze-draw stressed stability, the low pH and high pH stressed stability, and from the figure, it could be suggested that there's no change in CX purity and SHC purity. Then we conduct the high temperature stability study, and here are the results. From the left figure, we can see that after 6 weeks incubation at 40 degrees, there's a 3.5% increase of the low molecular weight species, which indicate a middle risk of fragment. And there's also a 10% mean peak increase and 25% increase of the acid peak variant, which suggests a high risk of charge variance. It could be suggested that the MAP-A is sensitive to the high temperature condition, and in the following CMC development, we will keep the protein away from the high temperature in the whole procedure. So this slide, this is a CMC development order from our client. According to the provided information, we found that the isoelectric point of the MAP-L is very low, which is only 6.4. So firstly, we conduct the homology modeling of the Fab region of the MAP-L. And the left figure is the charge surface profile. The color red is the negative charge, and the color blue is the positive charge. The right figure is the hydrophobic surface profile, and the color white is the hydrophobic patch. The color red is the hydrophilic patch. We could see that the MAP-L shows a negative charge patch in CDR loop, and there's no hydrophobic patch on surface. To further investigate its charge profile, we conduct a charge alignment between the MAP-L model with the reference model, and it shows that the local negative charge patch is significantly enhanced, which may increase the charge-charge repulsions inside the molecules and the interaction between the molecules, both of which may lead to unfolded population and protein aggregation. And in the following studies, the data also confirmed our hypothesis. As we see from the left bottom figure, there's persistent precipitation during process and stability study which suggests a high risk of high manufacturability. And from the other figure, we could see that after 4 weeks incubation at 40 degrees, there's an increase of aggregation for about 4%, an increase of low molecular weight species for about 6%, and the Cx mean peak shows a decrease of about 15%, all of which indicate a middle risk of aggregation, fragment, and charge variant. So we think we should refine this molecule. Firstly, we refined the cell line, and then we also added some projective agent in the process progress. Besides, we also optimized the projective agent ratio and its category. As we think from the results that the optimized MAP-L shows enhanced stability. From the picture, we can see that after 4-week incubation at 40 degrees, there is significantly no change of the fragment, the charge variant, and the aggregation. So in this study, we could see that the in silico evaluation method could help us to predict the degradation pathway of the molecules and lower the risk of the CMC development. In addition to this successful work, we also gained some useful experience from failure. From this study, we could see that the pre-preparation process of the samples is often neglected, but we found that if you didn't handle it well, sometimes a big problem may arise. In this study, we found that the stability of the sample X is very terrible. From the left picture, we can see that after 4-week incubation at 40 degrees, the sample X completely degraded. And from the other two figures, we can see that the mean peak percentage testing by SIEF shows over 50% reduction, which is a terribly high risk of charge variation. So we did the troubleshooting and we rule out the other probabilities, such as the testing errors or the instrument faults. We noticed that the sample is only purified by one-step purification. We suspected that there may some remained protease which could lead to the protein degradation. So we re-prepared the samples by 3 steps of purification, and the figures show that the stability is significantly enhanced. As shown in the left figure, the red line is the samples purified by 3 steps, and the blue one is the sample purified by 1 step. We could see that after 3-week incubation in different temperature conditions, the change of the mean peak percentage testing by SIEF is significantly reduced. And the most important is that at the 5 degree and 25 degree, there's significantly low change of the mean peak percentage. The same result was also shown in the right figure. So actually, the stability profile of this molecule should have been very good. Then after this case, in the following projects, we required samples with at least more than 2 steps of purification to prevent the false judgment. So all this above is our work in the antibody developability assessment. Lastly, I will introduce our company. Our ChemPartner Biologics is a wholly owned subsidiary of ChemPartner and established in 2015. We could provide Biologics CMC development and GMP manufacturing to support overall IND and NDA application requirements from our clients. Our one-stop service covers developability assessment, the stable cell line development, process development, and the pilot scale and large-scale GMP production. So if you have any questions or problems about today's topic or the CMC development, you can also contact me by the email address shown in this slide, and I will be very glad to discuss with you. So that's all for today. Thank you everyone for your time. We'd like to take some questions.
Well, thank you very much for that wonderfully insightful presentation. I would like to invite our audience to continue to send their questions or comments right now using the questions window for this Q&A portion of the webinar. Now, I've already received some questions for our speakers, so let's go ahead and take a look at our first question. Dr. Su, I believe this might be directed towards yourself. Right off the bat, we have someone wondering what kind of software will you use in the in silico evaluation?
Okay, it's a good question. So there are a lot of software for homology modeling of antibodies and developability evaluation. Usually we will use Schrodinger, Moe, Pymo, and to realize the in silico evaluation. But it also depends on the information and the requirements provided by our clients, because the developability assessment is usually highly personalized and case by case.
All right, very good insight. Thank you. Now, in the stability study, how do you select the buffer system of the candidate?
Okay. Firstly, it depends on the isoelectric point of the antibody. The pH value of the buffer should be away from the isoelectric point for about half to 1. According to this value, we will choose an appropriate buffer system to cover it. Thank you.
Oh, very good. Thank you very much for that. Now, I've got a question, I think, for Dr. Sun here. Someone would like to know, is there any way to know glycan modification on CRD region of the antibody?
Yeah, so this is a good question. So usually you could, for example, digest your antibody with IDEs and separate the generated species for the FC and the Fab to see whether your Fab domains that contain the CDRs exhibits a mass increase upon exposure the antibody to pathogen. So if this is the case, you could proceed by using additional proteinase to perform the peptide mapping, so which will allow you to determine the size of glycosylation.
Very good. Thank you for that. Next question for you would like to know, what is the LCMS characterization strategies for heterogeneous proteins?
Right. So usually for the highly heterogeneous proteins can be challenging to analyze by the intact mass LC mass spec. The complex spec more often difficult to deconvolute and interpret. So, however, it is often possible to remove common sources of heterogeneity, such as the in-liquid glycosylation or the C-terminal lysins previous to intact mass analysis, so that the other more pertinent or low-level features, such as the glycation, drug conjugation, or the heterodimer, the homodimer ratio of biospecific can be more easily detected.
All right, very good. Thank you. Now, would you say it's possible to keep an antibody bound on a protein AG SPR chip?
For the SPR technology, it's recently years is much more used in the characterization of the biopharmaceuticals. So typically, the regeneration conditions are determined especially because every type of the interaction is different. So the protein A or T binding to the antibodies is relatively strong, but can be regenerated. So for example, the glycine with the pH 2.7, if the interaction between our IgG of the interest and the analyte is also too strong, so the regeneration of the analyte alone will not be possible. So if the interaction is not that strong, you may find a regeneration condition which will dissociate the analyte without the dissociation, the antibody. So you will have to determine the regeneration condition for interaction pair empirically.
This is great. Thank you very much for that answer, and for both of you for all of your answers. However, we have reached the end of the Q&A portion of the webinar today. Now, if we couldn't attend to your questions, though, the team at ChemPartner will follow up with you, or if you have further questions, you can direct them to the email addresses that are up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I've also sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thanks for coming.
Thank you very much, Ray.
Thank you.